When we do things to change around the weak interactions that hold together secondary, tertiary, and quaternary structure in protein, we call that process denaturation. Notice that denaturation doesn't do anything to the primary structure of a protein. In other words, we're not affecting the peptide bonds in a protein, but we are affecting all those other weaker interactions that we saw going on in previous slides. So we're potentially destroying the secondary, tertiary, and quaternary structure of a protein. So in denaturation, we take those weak interactions and we disrupt them. And if we do that in a permanent way, as for example happens in the albumin protein of a fried egg, then we end up with a permanently denatured protein. We permanently change the structure of the albumin, and it can't go back to the structure that it had before. A fried egg doesn't unfry itself no matter how long we wait or no matter what we do to it. In some cases, we can denature in what's called a reversible way, and so the protein can fold back up into its normal configuration even after we've disrupted these secondary, tertiary, and quaternary interactions. This is what happens, for example, when we warm milk and then cool it back down again. So the proteins in milk will denature, but then they're able to spontaneously renature back into what scientists call their native forms, the forms that they had before we started the heating process. So as I mentioned earlier, we can take the protein which is found in egg whites called albumin, and we can denature it in a couple of different ways. We can denature it in a hot pan, and so it's called frying an egg. So if we fry an egg, we change the relatively clear and liquid albumin solution into a white and not very liquid but rather solid fried egg. So this becomes the white of the fried egg. This is just from the denaturation in an irreversible way of the albumin protein. Or we can separate out those eggs. We don't want the yolks when we do this. We want just the whites. And with just the egg whites, in other words, an almost pure albumin solution in water, we can denature it with a whip. And by whipping those egg whites, we can turn it into a meringue that we might use on top of a pie. Another example of denaturation which we see in the kitchen is the protein collagen. Collagen is the main structural protein of the body, so we find this in almost all the connective tissues of the body. The primary structure here is shown by the amino acids linked together. The secondary structure is sort of disorganized, and so it's neither an alpha helix nor a beta pleated sheet, but the tertiary structure is a long strand with red, green, and blue strands, also twisted together into a triple helix, which gives this a lot of strength. If we denature this, we turn this from oriented strands, which is what we find in the native form inside your body, or in this case inside the pig's body. We can use denaturation to turn those native strands into denatured strands. Notice denature is the removal of the native configuration. So denaturation turns connective tissue into gelatin. And just as a little aside here, not something you need to learn, but just something that I thought was interesting, is about not quite half of our collagen that we use in Jell-O comes from pig skin. About a quarter comes from cow hides, and about a quarter comes from bones. The reason that we spend so much time on this is that proteins are a very, very important part of the human body, and proteins are what allow all the different functions of the human body to be carried out. For example, we've looked at collagen as a structural protein. We've talked briefly about insulin as a signaling molecule. Proteins named actin, troponin, and tropomyosin are used for contracting muscle cells. A protein called immunoglobulin G is used to protect the body from invaders. Myoglobin is an oxygen carrier in muscle, the same way that hemoglobin is an oxygen carrier in blood. And carboxypeptidase A is an enzyme. We'll look at enzymes in the next couple of objectives in just a minute.